So welcome to today's video. In today's video, we are going to discuss about the stellar evolution in pteridophytes. In previous video sessions, we had discussed about the general characters of pteridophytes. If you had missed that video, please do watch it and follow it up. And in today's video, we are going to discuss about stellar evolution. So before diving into the topic, I would like to give a brief explanation or introduction about the today's topic. So stellar or evolution. The word itself says stelar. The word stele is a Greek word which means pillar or column. So we would have studied about the vascular bundles, that is xylem and phloem. So the xylem and phloem, they are arranged collectively and they are called as stele. So it is called as stele in case of pteridophytes. In angiosperms and gymnosperms, we had called it as vascular bundles, but in pteridophytes, we call them as stele. So a stele represents the entire unit of vascular bundles. So we had studied about algae, bryophytes and all. They were those plants where they didn't have any vasculature. So these pteridophytes are the first plants to develop the vasculature. Vasculature in the sense I mean here about the xylem tissues and the phloem tissues. We all know xylem, it is used for water conducting purpose, and phloem, it is used for food transportation and mineral transportation or translocation we call it. So the stele is a Greek word which means pillar or column. So stele is the central axis of primary vasculature which includes xylem, phloem and the surrounding tissues around the vasculature such as pericycle, pith and endodermis. If you take any structure, primary structure of angiosperm or gymnosperm, we find the central pith region surrounding which is the xylem, surrounding which is the phloem, surrounding it is the pericycle and surrounding it is the endodermis. So some scientists say that endodermis is the outermost layer of the stele, while some other scientists say that endodermis is the innermost layer or limiting layer of cortex. So if you see, you have epidermis, you have cortical region or cortex region, and the stele is over here. So this will be your endodermis and pericycle, and the stele will be present here. So they say that endodermis is the innermost layer or limiting layer of cortical region. Some scientists say. Some other include endodermis as the outermost layer of the stele. So generally when you say about stele, stele is a primary axis which includes the primary vascular structures such as xylem, phloem and surrounding tissues such as pith, pericycle and endodermis. So this is stele. So whenever you hear the word stele, you should get pith, xylem, phloem, pericycle and endodermis. So these things should be clear. So in pteridophytes, if you see, stele can be broadly classified into two parts. That is protostele. Proto means first or primitive. So in pteridophytes, until bryophytes, there was no stele or no vasculature. For the first time in the pteridophytes, the primitive members like xylotales, xylophytales, lycopodales, selaginellales, you had protostele. These are primitive stele. So in this protostele, what do you mean by protostele? So protostele is a type of stele where pith is absent. So pith is the central part of the vasculature. Pith is also called as medulla. Pith is also called as medulla. So in protostele, pith is absent, that is medulla is absent. Hence it is also called as non-medulated stele. Hence it is also called as non-medulated stele. So medulla in the sense pith. So that is the characteristic feature of protostele. We have second division called as siphonostele. In siphonostele, the pith or medulla is present. Hence it is also called as medulated stele. This is called as non-medulated stele because medulla is absent or pith is absent. Protostele is called as non-medulated stele. And siphonostele is called as medulated stele because pith is present. Siphon. Siphon means a hollow cylinder or a hollow pipe. Center will be hollow. That is what pith. Pith can be hollow cavity or it can be made up of parenchymatous cells or it can be made up of sclerenchymatous cells. So there is in the central pith is present in case of siphonostele. So we will study what and all are the types that comes under Protostele and Siphonostele. How did steles evolve from the most primitive type to the most advanced type? We will see the evolutionary changes also. So under protostele, we have major 5 types: haplostelic protostele or haplostele, actinostele, plectostele, mixed protostele, and polystele. So what is haplostelic protostele? Haplostelic protostele is the most primitive type or most simple type, where you have xylem. Surrounding the xylem is phloem tissue, that is the food conducting tissue. Surrounding the phloem is pericycle, and surrounding this is endodermis. So above you have cortex, epidermis. We're not dealing about it. We're dealing only about the stellar region. So the centermost you have xylem, next followed by phloem, followed by pericycle and endodermis. Since it is a type of protostele, pericycle is pith is absent here. So this is the thing. And in xylem, we all know metaxylem and protoxylem. So protoxylem they are formed first and metaxylem they are formed later. So depending upon the number of protoxylem points, we classify them as monarch or diarch, triarch, tetrarch or polyarch. If there is one protoxylem, then we call them as, so here you can see there is only one protoxylem. If it is arranged in this manner, it is called as monarch. If there are 2 protoxylem, then they are called as diarch. Similarly, based on that, if there are 3, then they are called as triarch. So based on the number of points, they are called as monarch, diarch, triarch, tetrarch or polyarch condition. So, and also, what if the protoxylem is present towards the center and metaxylem are present towards the periphery? So such a condition is called as mesarch. We all know meso means between or center. So mesarch is a condition where the protoxylem are towards the center, surrounding them are your metaxylem. So this is called as a mesarch. If the protoxylem are towards the periphery, then they are called as exarch condition. If the protoxylem are towards the periphery or facing the epidermis, they are called as exarch condition. If the protoxylem is towards the pith, here there is no pith, generally we refer them as pith. If the protoxylem is towards the pith or if they are away from the epidermis, if they are facing inside, they are called as endarch. So if they are like this, so we denote protoxylem in a smaller lumen with metaxylem broader lumen. So if the protoxylem is away from the epidermis, then we call them as endarch. This is called as endarch condition. This is the basics that we have to know when we are studying about a stele or any vasculature. If the protoxylem is here and the metaxylem is the other way, so protoxylem is facing towards the epidermis, it is facing outside. That is called as exarch. If the protoxylem is in between, surrounding which is the metaxylem, then such a condition is called as mesarch. So these are 3 types of xylem points. So based on this, the haplostele can be further classified or further broken down into monarch or diarch or triarch or tetrarch. So this is about haplostele. I hope this much is clear. So the second point is actinostele. Second type under protostele is actinostele. Actinostele, here what happens, the xylem rays. So if this is the xylem rays as I drew over here, these are called as xylem rays. So the xylem strands, they get broken down or they get branched. They get branched and they appear like radiating arms. They appear like radiating arms. So such type, so in the stele you appear like this. In the stele the xylem appears like this, like radiating arms. They are furrowed or they are deeply furrowed or branched to form radiating arms. Each arm, like they appear like star-like, stellate they are also called as. So star-shaped xylem or stellate xylem is called as actinostele. In between these you will find phloem. In between these radiating arms, you will find phloem. So actinostele is very simple, star-shaped or stellate xylem, where the xylem is furrowed into radiating arms. In between radiating arms or alternating with radiating arms, you find patches of phloem alternating with radiating arms. Such type of stele. So surrounding this again will be pericycle, surrounding this will be endodermis. This is common. Pith is absent. So such type of condition is called as actinostele, stellate shaped or star shaped. It is seen in case of Lycopodium species. As I mentioned, it is most primitive type when compared to Siphonostele. So you can find the examples from the lycopods or from selaginals, like xylotales. From the primitive members of pteridophytes, you can find them. That is actinostele. Next comes your plectostele. Plectostele, the furrows get still more deepened and they form plate-like structures. So the furrows are still more deepened and they form plate-like structures. So this xylem forms plate concave disc shaped or plate like structures. Such type of stele is called as plectostele, and surrounding each plate will be phloem. Surrounding each plate you can find the phloem. In center you can find the xylem. So you have, for example, this is a single plate and this is your xylem, surrounding which you will find phloem. Above which another plate will be there, surrounding that you will have phloem. Above which another plate will be there, surrounding that you will have phloem. So they are arranged plate-like, hence they are called as plectostele. It is seen in Lycopodium whiteanum species. Lycopodium whiteanum stem section, if you take, you can find such type of arrangement. Whereas in Lycopodium phlegmaria, you can see actinostele that I discussed before, stellate shaped xylem. So this is about plectostele and this is about actinostele. Mixed protostele. Under mixed protostele, what happens? The plates gets broken down and they are scattered into bits. So the plates gets broken down and they're scattered. So here there will be xylem, here there will be xylem, here there will be xylem. So these are all the broken bits of the plates surrounding which the phloem is present. So xylem surrounding which the phloem is present, they are broken down into bits. The plates are still more deepened, furrowed, and they get broken down into smaller bits. So these such type of arrangement surrounding this will be pericycle and endodermis. That's common. So such type of thing, they are mixed, right? So hence they are called as mixed protostele. And if you have many rings of haplostele, it is called as polycyclic protostele. Poly means many, multiple. Cyclic means they are arranged in a cyclic manner. So in a stele, if you find many haplostelic protostele, such type of stele is called as polycyclic protostele. So I hope protostele is clear. So under protostele, it is also called as non-medulated protostele or non-medulated stele, since the pith is absent. And under that we have 5 types: aplostelic protostele, actinostelic protostele, plectostelic protostele, mixed protostele, and polycyclic protostele. So these are the 5 types under protostele. Next, when we come for siphonostele. Siphon, as I said, means it is a hollow cavity. So here you have pith. How did pith develop? So long we studied the xylem, phloem, pericycle, and endodermis and cortex you had, and you had epidermis. So there was no pith. Suddenly, how did pith develop in the course of evolution? To study that, you have 2 theories. The first theory is called as expansion theory. It is called as expansion theory. I will give you why it is called as expansion theory. So what, according to this theory, it suggests that those xylem that are present in the center, they gave rise to cells. We know xylem is a living tissue. So they will differentiate, they will duplicate, they will divide. So xylem, it cuts off cells and it gives in between more cells. So the xylem, it cuts off and it gave rise to many cells over here. These cells, on the course of evolution, they became parenchymatous and they became pith. So the xylem tissues that were there in the center, it gave rise to many parenchymatous like cells on the course of evolution, which they became parenchymatous and they gave rise to pith. They were called as pith. So when the central region, the pith developed, xylem expanded. It's like how the balloon is expanding. The central region, the pith develops. So as the pith develops, it enlarges. So the xylem also expands. So hence the name expansion theory. And the origin of the pith is xylem cells. Who gave rise to pith? Xylem gave rise to pith. Xylem is a part of stele. As I said in the introduction, stele consists of xylem, phloem, pericycle, endodermis, pith. Xylem is a part of stele. One of the member of stele gave rise to pith. Hence, it is also called as intrastellar origin of pith. Intrastellar origin of pith. Intra means within. So stellar. So the stele, one of the member of stele. Intra gave means one of the member within. Within the stele, one of the member gave rise to pith. Hence, it is also called as the same expansion theory is also called as intrastellar origin. So I hope it is clear. Expansion theory in the sense the xylem gives rise to more cells that becomes parenchymatous, and the course of evolution they became pith. And xylem also expands. Hence the name expansion theory. And xylem is one of the member of the stele. Hence it is also called as intrastellar origin. This is one hypothesis. So here the first theory, xylem is the origin. So from the how did pith come? Xylem is the origin of the pith. The second theory is called as invasion theory. Invasion theory. So what is this invasion theory? According to this theory, scientists say that the cortical region is made up of parenchymatous or sclerenchymatous. We all know that this cortical region invaded through the leaf gaps. They invaded through the leaf gaps or the branch traces, leaf traces. We all know leaf gaps are nothing but the stele breaks or branches open and it gives rise to vascular bundle to the leaves. So that is called as leaf gap. So if this is the stem and here you can see the steles that are arranged. You can see the stele, central stele. This branches over here and it gives rise to stele for this leaf. So there's a branch that is called as leaf gap or leaf trace or branch trace. So the cortical cells or the cortex region invaded through those leaf gaps and branch gaps and they formed pith. Since there is invading or invasion, this theory is called as invasion theory, and it is also called as extrastellar origin. Extrastellar. It is not part of stele. The cortex is not part of stele. Only xylem, phloem, pericycle, endodermis and pith is the part of stele. Cortex and epidermis are outside the stele, extrastellar. So they are not part of stele. So hence this theory is also called as extrastellar origin of pith. So what do they say? Invasion theory. The cortical cells invade through the leaf gaps and branch gaps, traces, leaf traces and branch traces, and they form the parenchymatous pith. So these are the two theories that say that Protostele on the course of evolution developed pith. And also if you see the expansion theory, that xylem is the origin of the pith, it is seen in primitive members. And in invasion theory, where the cortical cells invaded and it formed pith, it is seen in higher members of pteridophytes. In case of ferns and water ferns like Marsilia, Salvini, Azole and all, invasion theory is proved. In lower forms like Lycopodium selaginella, Equisetum and all, we can find expansion theory to be valid. So these are the two theories that are proposed to show or to explain how pith was developed. So under Siphonostele, we have 6 types: ectofloic Siphonostele, amphifloic Siphonostele, solenostele, dictyostele, eustele and atactostele. We go to ectofloic Siphonostele. Ectofloic Siphonostele is the most primitive type of Siphonostele, where you have central pith. As I said, it may be parenchymatous, sclerenchymatous or hollow cavity, surrounding which you have xylem, water conducting tissue, surrounding it you have phloem, food conducting tissue, surrounding it you have pericycle, and surrounding the pericycle you have endodermis. So the pith is present. This is similar to that of haplostelic protostele. Only difference is in protostele there is no pith. In Siphonostele pith is there. Rest all remains the same. This is ectofloic siphonostele. Ectofloic siphonostele. Second type is amphifloic siphonostele. Amphi means double or dual. Bryophytes are called as amphibians of plant kingdom because they need both water as well as land, both dual nature. So amphi means both. Amphifloic siphonostele. So what is amphifloic siphonostele? In amphifloic siphonostele there is central pith, and the pith is surrounded by xylem. Pith, the central pith, pith is surrounded by xylem. Xylem in turn is surrounded on both inner as well as outer side by phloem, pericycle, endodermis. So you have pith, surrounding pith you have xylem. Xylem in turn, both inner side as well as outer side is surrounded by phloem, endodermis, pericycle. So you have two phloem layers, two pericycle layers, two endodermis layer, one on either side of xylem. One will be inner side, one will be outer side. So pith is there, surrounding which xylem is there. So surrounding the xylem you have, towards outer side you have outer phloem, towards inner side you have inner phloem. I said no, dual nature, double thing, outer phloem, inner phloem. Surrounding outer phloem you have outer pericycle, and surrounding this inner phloem you have inner pericycle. Surrounding outer pericycle you have outer endodermis. Surrounding this inner pericycle you have inner endodermis. So you can find two layers, one outer layer, one inner layer. So this, if you see the order of arrangement, you have central pith, surrounding which is the inner endodermis, inner pericycle, inner phloem, xylem, outer phloem, outer pericycle, outer endodermis. Such type of arrangement is called as amphifloic siphonostele. Amphi means double. Amphifloic siphonostele. So you have to remember this amphifloic siphonostele. Later also we will be discussing. Again, amphifloic solenostele we will be discussing. So amphi means double. You have inner phloem, outer phloem, inner pericycle, outer pericycle, inner endodermis, outer endodermis. You have to remember that much. Xylem is only one layer. So this is the first two types, which is most important type, and it is seen in majority of the cases. And amphifloic, that I said, no, inner outer, that is seen in Marsilia rhizome tears. When you take the rhizome section of Marsilia, that is water fern, you can find this amphifloic condition. Next we find solenostele. What is solenostele? So you will have a central pith, surrounding which is a xylem, surrounding which is the phloem, surrounding which is the pericycle, surrounding which is the endodermis. This is normal siphonostele. If the normal siphonostele is traversed by a leaf gap, it is, so this becomes separated. This becomes one and this becomes one. So this is your leaf gap. So this becomes a leaf trace. You have to remember leaf gap and leaf trace. This is a gap. So you have leaf gap. This is a leaf trace. So this gets branched and this part goes to the leaf. So this is the main stem. From the main stem the vascular bundle is broken, and this leaf gap vascular bundle, it traverses to the leaf. That is called as leaf gap and leaf trace. So if the siphonostele is traversed by a single leaf gap, then it is called as solenostele. I am stressing on the word single leaf gap. So there will be only one leaf trace or single leaf gap or single leaf trace. If it is traversed by a single leaf trace, such type of stele is called as solenostele. Under solenostele, again you have two types, ectofloic and amphifloic. Ectofloic solenostele, pith, xylem, phloem, pericycle, endodermis. Similar to that of ectofloic siphonostele. And you have amphifloic solenostele also. Again in amphifloic also you have only one leaf gap. But only difference is you have inner phloem, outer phloem, inner pericycle, outer pericycle, inner endodermis, outer endodermis. So again this solenostele, it is traversed by a single leaf gap. It is further divided into ectofloic and amphifloic. So this is about your solenostele. I hope you all understood this. Only one leaf gap will be there. If this is broken by many leaf gaps, if this is broken by many leaf gaps, then such type of stele is called as dictyostele. Then you will have many parts. This will become one unit, this will become one unit, so this will become one unit, right? This is first unit. Similarly, this will become another unit. This will become another unit. This will become another unit. So each unit is called as meristele. Each unit is called as meristele. And such type of condition is called as dictyostele. So understand this. You have the central stele, same with pith is present and siphonostele. If they are broken, it's like a single cake being cut into many pieces. If a cake is cut into one piece, then it is called as solenostele. If cake is cut into multiple pieces, it is called as dictyostele, and each piece of cake you serve is called as meristele. It is like a pie cake. So you can find single cake over here. If you cut a single piece, then it is called as solenostele. If you cut into many pieces and single piece of cake is called as meristele. Similarly, the single unit, discrete unit of this dictyostele is called as meristele. This is very important. How the meristeles are arranged, based on that there are further classifications of siphonostele. So I hope you understood the major type: ectofloic siphonostele, amphifloic siphonostele, and solenostele. Only one leaf gap, that is further classified into ectofloic and amphifloic. And dictyostele, many leaf gap, and each unit is called as meristeles. Next we find eustele. So it is based on the arrangement of meristeles. So if the meristeles are arranged in a concentric rings and they are overlapping, if the leaf gaps are parallel, there are many leaf gaps. If the leaf gaps are parallel, what happens? If the leaf gaps are not parallel, one meristele will be here, the other meristele will be here. It won't overlap. It will be in between these two. Since the leaf gap is parallel, you have overlapping meristeles. So you have overlapping meristeles. Such type of stele is called as eustele. Such type of stele is called as eustele, and it is found in dicotyledons. So I will repeat again. If the meristeles are overlapping and they are arranged in a concentric ring, these are meristeles, these are overlapping, and these are arranged in concentric rings. Such type is called as dicotyledonous type or eustele, which they call it as conjoint collateral steles or vascular bundles. So such type is called as eustele. If they are randomly scattered, they are not arranged, they are randomly scattered here and there in the ground tissue, then we call them as atactostele. Atactostele. This is seen in case of your monocotyledons. So when you take monocot section, the vascular bundles are scattered in the ground tissue. There is no differentiation with cortex and pith region. Same condition is seen over here. The steles are scattered in the ground tissue and there is no connection or no distinction between cortex and pith. And such type of scattered stele condition is called as atactostele. Is called as atactostele. So under siphonostele we have ectofloic, amphifloic, solenostele, dictyostele, eustele, and atactostele. So these are the types you can see, and all these types is seen in case of pteridophytes. That is most astonishing. For example, if we take any pteridophyte, let it be Xylotum or Selaginella, the root will be having one type, the rhizome will be having another type, the aerial shoot will be having another type, old shoot will be having another type of stele. So in a single plant or collectively the entire pteridophytes will have the entire all types of protostele can be seen. Hence this is very important to study the stellar evolution. How did it evolve? So that is how what we are going to discuss now. Which stele was there came first? From which what stele evolved? From where it became? How did the simple primitive protostele become most complex stele? That is what we are going to study now. That is the last topic of thing, evolution of all the steles. So you have to know the basic idea of thing. So there was a parent line, that is your haplostelic protostele. Haplostelic protostele. This is the most primitive thing, as I said. The haplostelic protostele, it branched and gave three lines, three evolutionary lines. First line, it became actinostele. So all this line, it won't have any pith at all. So all these are non-medulated type only. The protostele, it branched and it gave into radiating arms, stellate like star shaped, it became star shaped and it became actinostele. This star shape became, the grooves became more deepened and it became plate like to form plectostele. So you can see the evolution has, how evolution is occurring. So the complexity is increasing. The most primitive protostele gave rise to actinostele. Actinostele gave rise to plectostele. Plectostele gave rise to the most evolved complex type called as mixed protostele. Mixed protostele, because the plates are broken and they are dispersed or scattered. So it is mixed protostele. This is first line of evolution. There are no pith anywhere. It is called as non-medulated stele. That is nothing but these are all the types of protostele. In the second line of evolution, it gave rise to amphifloic siphonostele. Amphifloic siphonostele. Pith developed by means of two theories: expansion theory, invasion theory. Amphifloic siphonostele, and on the other line it gave rise to ectofloic siphonostele. Ectofloic siphonostele. This amphifloic siphonostele gave rise to amphifloic solenostele. I said it right, solenostele, only one leaf gap. It is further divided into ectofloic and amphifloic. This amphifloic siphonostele gave rise to amphifloic solenostele. And this amphifloic solenostele gave rise to dictyostele. Dictyostele. So dictyostele, and dictyostele gave rise to polycyclic stele. Polycyclic stele. So you have many cycles over there. So I hope this line is understood. Haplostelic protostele gave rise to amphifloic siphonostele. Amphifloic siphonostele gave rise to amphifloic solenostele. Amphifloic solenostele gave rise to dictyostele. Dictyostele gave rise to polycyclic stele. Dictyostele, it is traversed by many leaf gaps. Next, ectofloic siphonostele gave rise to ectofloic solenostele. Ectofloic solenostele gave rise to U-stele. U-stele, as I said, dicotyledons. I mentioned about dicotyledons where they are arranged in a conjoint collateral manner. The leaf gaps are overlapping because of the parallel leaf traces. The vascular bundles are overlapping. Such condition, such a stelic condition is called as U-stelic condition. So, ectofloic solenostele gave rise to U-stele, where the steles are arranged in conjoint collateral manner. They are arranged in concentric manner. If this U-stele gave rise to atactostele. Atacto, they are irregularly arranged. They are scattered in the ground tissue. So atactostele is the most complex stele in this line of evolution. Polycyclic stele is the most complex or most advanced. Mixed protostele is most advanced. So this is the most primitive type that you can see in the primitive members. So this is how the evolution has occurred, and you can see this in monocots and dicots, and this you can see in higher pteridophyte members. So this is all about stelar evolution. You have to just remember the types of protostele and siphonostele. And a degree university examination, it can be a 5 marker or a 10 marker question, long length questions, or 2 marks. They may ask like what is actinostele, what is any one type, what is siphonostele, what is medulated stele, or they may ask expansion theory, invasion theory also for 2 markers. And this is the stelar evolution flow chart which you have to remember. This is very important in case further study. So when you're studying pteridophytes or the type study, they will just mention you the stele is actostelic or siphonostelic, solenostelic. So you have to know what stele is which stele. So thank you for your patience listening. If you like this video, please like my channel, subscribe my channel, and comment your valuable comments and share this video through your friends. And apart from this, 2 more videos have been put forth. That is a general characters of pteridophytes, where in detailed description of the pteridophytes, almost from 25 to 30 points has been explained very thoroughly. And also in another physiological cycle called as glyoxylate pathway, which is very important pathway, has been discussed in my videos, in my previous videos. Please do watch that, like, subscribe and comment. Thank you.